Okay, pleasure to welcome Luke here. Luke has just started his postdoc at UC Davis in the Centre for Population Biology. For that, he started as a dinosaur hunter at the University of Chicago. Then went to the Field Museum and then to WashU and translocated with the Los Altos lab to Harvard. He did his PhD, and as I said, he's now at Davis. So welcome, Luke. Good to hear about your work.
Thanks a lot, Craig. Charles.
Someone should teach you well, so leading really personally.
You lied again, Marshall.
Yeah, always thanks. Yes.
It's definitely a real honor to be at the MVZ lunch. I spent 7 years as a PhD student hearing legends about the MVZ lunch from my advisor every year. So yeah, it's a real honor. So I'd like to start off today by talking about my interests sort of at a broad scale. As long as I've been interested in biology, which has been, I think, as long as I can remember, since I was crawling around, two things have sort of captivated me. I've always been interested in, on one hand, ecological interactions. So, you know, why two different species of lizard, for example, might compete for the same perch, or you know, how a host might deal with its parasite load. And on the other hand, I've been interested in phenotypic diversity. And you know, as early as I can remember, I've been, you know, I was sort of an outdoor kid and had field guides and a creek nearby and would catch a lot of things. And I was always interested in essentially the diversity of things, how they got to be so different, why they were different, whether that had anything to do with their ecology. You know, for the last several years, as I've tried to develop a research program, it's really the interaction between these two interests that have driven my research. So I'm more or less interested in how ecological interactions influence the evolution of phenotypic diversity. And the scale at which I'm interested in this is a very macroevolutionary scale. And so I'm interested in answering the question of how do ecological interactions affect broad-scale patterns of evolution? And do they leave signatures over macroevolutionary time? And so during my PhD, I did a number of studies in which I addressed different elements of this broad question. And today I'd like to talk about two. The first one is an exploration of the ecological opportunity hypothesis. And in this study, I'm really asking, does competition, you know, one type of ecological interaction, influence the rate of phenotypic evolution, the rate of diversification in form? And then for the second part, I'll switch gears a little bit, and I'll ask how ecological interactions influence evolutionary pattern rather than rate. And here, what I'm doing is, and I'm really excited actually to talk about this. It's sort of a different kind of talk, but I'll talk about a species discovery that I think has implications for our understanding of the role of ecology in shaping convergent evolution in a classic example of replicated adaptive radiation. Before I get started with that stuff, though, I figure I'll spend at least a minute introducing my study system. And so I work on Anolis lizards, and so lizards of the genus Anolis are, I think if anyone's counting, it's the most diverse amniote genus. There was a recent split might be the most diverse terrestrial vertebrate, since they recently split up the Lutherodactylus into different genera. But to the uninitiated, anoles are small to medium-sized lizards. They're neotropical, and they're really known for their arboreal speciality. They've diversified into arboreal niches, and that's where you usually find anoles. So you can find them in the trees. Now, what anoles are, of course, most famous for is carving up the arboreal habitat into different types of microhabitats. You know, some of them are large and are specialized for canopy life. Some are gracile, have long tails. They are almost exclusively found in grasses and bushes. But they're found throughout the neotropics. And the way you know you've found an anole, usually, almost always, it'll have a combination of two traits that are fairly anole specific. First, they'll have a dewlap. They have an extensible flap of skin that they use for communication. And second, toe pads. And like geckos, anoles have adhesive toe pads. They evolved them convergently to geckos. And they you have lamellae, which have microfilamentous CD, which they use to cling to vertical substrates. So if it's got a dewlap and it's got toe pads, it's an anole. Not all anoles have dewlaps, most do. Not all anoles have toe pads, there are two that don't. But for the most part, that's how you tell an anole. So if you're interested in ecology and diversification, you might ask why study anoles. Actually, most people here probably wouldn't ask that, but if you're wondering, I think there are a lot of reasons why anoles make a great study system for diversification. And I'll point out really quickly, today I'll just be talking about anoles in the Greater Antilles, which I think are a particularly good system for studying diversification. So reasons you might study anoles, first of all, they're extremely diverse. So if you're interested in diversification dynamics, it helps to have a group that has a lot of diversity. And anoles are diverse in species number, they're diverse in their ecological roles, and they're also diverse in morphology. And so you can ask a lot of different questions about the evolution of diversity in anoles. So there are nearly 400 species, and there will certainly be 400 species at some point. In addition, the systematics of the anoles radiation, at least relative to other lizard radiations, is relatively well known. So especially for the Caribbean, we have pretty sound understanding of the phylogenetic relationships. Nearly all of the species are represented in a single phylogenetic hypothesis. And we're fairly confident that we know about most of the species, at least in the Caribbean. So that's helpful. And really key to my interests is that anoles have diversified in a variety of conditions, under a variety of conditions. And so anoles have radiated numerous times in numerous different settings. And so Caribbean anoles, almost any rock that you can find in the Caribbean will have some anole species. If the rock is the size of this room or larger, it's virtually guaranteed. So anoles occur in a wide range of ecological communities, from small single species islands up to regional type islands like Cuba, where you get upwards of 60 species. What's really key is that on the 4 largest islands in the Caribbean, the Greater Antilles, you actually have diversification occurring within islands, and it's occurred at different times. As you can see in this phylogeny, what I've done is we've got Cuba in red, Jamaica in green. Those two colors are obvious. We have Hispaniola in yellow, and then Puerto Rico in gray. And here's the timing of diversification and the geography of diversification throughout the Greater Antilles. I should say the relative timing. I'm not actually providing an absolute date because there's some contention, but we've estimated rates of diversification, assuming a common fixed point. What you can see is that diversification is, first of all, largely occurring within islands. So, this is a large, actually one paraphyletic Hispaniolan radiation there. But occasionally we get colonization from one island to the next, followed by subsequent diversification. And so diversification is occurring. There are 2 different types of scenarios. So for example, when it kicks off here in Jamaica, you'll notice that Jamaica is represented only in a single place in the tree. It's a monophyletic radiation. There's probably very high ecological opportunity at the base of the Jamaican radiation. However, we have parts of the phylogeny where ecological opportunity is probably very low. And so, so we're towards the tail end. The Cuban radiation, we have speciation events that are occurring when there are already dozens of species on that island. So a big diversity of ecological settings where diversification is taking place. Equally important, and maybe even most important, the anoles provide a strong foundation of studies that can help you link ecology to evolution. So there have been decades of work on the ecology and evolution, or the ecology of anoles. So when we're looking at trait evolution and patterns of evolution, we have a firm understanding of the ecological significance of these evolutionary changes. So for example, the relationship between habitat use, morphology, and behavior is fairly well known for a fairly large sample of anoles. So this helps us to interpret large-scale patterns of evolution. So to move on to the first project, I was interested essentially in looking at whether the signature of competition, one type of ecological interaction, has a macroevolutionary, whether that interaction has a macroevolutionary signature. So I'll start by talking a little bit about adaptive radiation. Adaptive radiation is gotten a lot of attention recently, and the idea is that the ecological theory of adaptive radiation posits an ecological explanation for large-scale diversity patterns, and really that ecology does influence macroevolution. When we think about adaptive radiations, we think about these bursts of diversification, like the honey creepers of Hawaii, where you have this unoccupied set of niches, and then a lineage reaches it and then adaptively radiates into a bunch of different niches. What's key, I think, is the concept of ecological opportunity. Ecological opportunity has been in the literature for a long time, but it's pretty tough. It's one of these slippery words that I think is tough to define. So I'm gonna stick with a definition that proposed it as a provisional definition that Schluter proposed in 2002 or 2000. Which is a wealth of evolutionarily accessible resources, little used by competing taxa. And Schluter really, what I like about this definition is that it focuses on competition, and that's something that we can at least try to get an estimate of, that we can take a step towards measuring. So from this we can formulate what people are calling the ecological opportunity hypothesis, which is that the pace of adaptive diversification over large time scales is regulated by competition. And the prediction is that as the number of competing lineages increases, the pace of adaptive diversification should decline, and adaptive radiation should wane. It should be diversity dependent. Now, the first people to be interested in testing these predictions, asking these questions, people have been interested in this, I think, at least since Simpson in the 1950s, this theory of adaptive radiation. It's always been somewhat of a difficult hypothesis to test, and the first folks to really take a crack at it were quantitative paleontologists in the 1970s. So I think at that time, paleontologists were really the only people with the appropriate view to look at macroevolutionary scale questions. Recently, however, people have started using molecular phylogenies to ask macroevolutionary questions, and there's been a huge surge of interest in looking for diversity-dependent patterns in lineage diversification using molecular phylogenies. And the inference is fairly simple. Under an expectation where, under a model where there is no ecological, there is no ecological process affecting diversification, you would expect exponential diversification. So it's a linear increase on a semi-log plot of lineages through time, if lineages are radiating adaptively, if they're ecologically diversifying. So here you can see this is just a pure birth phylogeny. This is, of course, from a paper, Charles Marshall's and Cago-Kentel. Now, if diversification is diversity dependent, you would expect a burst when the ecological opportunity is higher, when there are fewer species, and then a slowdown. And so, if you plot the same semi-log plot, you would expect a convex pattern of lineage accumulation. And this is usually represented by a negative gamma statistic. So there have been a lot of studies that have used these methods, looked at lineage diversification over time to infer adaptive radiation and test ecological hypotheses deep in the past. What I would argue is that it's important to remember that the hypothesis really is about adaptive evolution, and I would argue that it's valuable to complement these studies with studies of trait evolution and really ask whether the pace of adaptive evolution, adaptive trait evolution, is showing the same pattern. I could think of a lot of reasons why lineage diversification might exhibit this convex accumulation pattern that may have nothing to do with ecology or adaptation, for example, at least not species interactions. Maybe diversification along an archipelago might have sort of a burst under that situation. And so I think diversification at adaptive traits is somewhat key. Darwin's finches are not famous because there are tons and tons of lineages, but because they're adaptively diverse. So the question I'll address is how ecological opportunity influences morphological evolution. Now, for lizards, the prediction is that when there are fewer lineages around, you can imagine diversification occurring let's look at the brown anole in each example. Say we have two different islands, and diversification is occurring in this focal lineage. We would predict that the rate of evolution is going to be higher when there are fewer competitors around. Fairly simple. So in order to test this, we need to do two things. We need to estimate historical ecological opportunity, and then we need to estimate rates of evolution or measure rates of evolution. And then we need to test whether there's a relationship. And so I'll do this at the nodes above phylogeny. So both types of data are going to be node estimates, both ecological opportunity and the rate. And then I'll use a model comparison framework to look for a relationship between the two. So the data that I gathered for this project, and this is really sort of the centerpiece data set for my thesis. I've used it for a lot of other things. So I sampled as many species as was possible for the Greater Antilles, measuring both morphology and collecting tissues to build the phylogeny, and was able to sample 100 of 120 species in the Greater Antilles. Those we weren't able to sample are mostly cryptic species, and they're from, a lot of them are from Cuba, for example, were only recently described within the last couple of years. So it's hard to get samples for some of those, but I'm reasonably confident that I've managed to represent the lineage and ecological diversity in the radiation fairly well. I conducted several phylogenies, but for this project we have a phylogeny that's based on mitochondrial DNA, and we've created a chronogram, like I said earlier, with a relative origin. We don't have much to say about the absolute timing of diversification, but we did create a chronogram to look at the relative timing of anole diversification. And then for morphology, I measured basically as many traits as I could on as many specimens as I could in order to get a holistic view of morphological variation in anoles. And so these are external measurements, but the measurements correspond to underlying skeletal morphology. And so I won't go through the exact traits just because of time, but 22 traits from over 700 specimens. I log transformed the traits, I size corrected them, and then I performed phylogenetic principal component analysis. And just to sort of give you the short of it, I have a SVL treated separately as a size axis, so body size is one of the key traits. And then I retained 4 shape principal components after my principal component analysis. And so PC1 is limb length, PC2 is head shape, PC3 is tail length, which is quite variable on anoles, and 4 is topad lamella number. Now, when I measured characters, I was sure to measure characters of classic ecomorphological importance, those that have been shown to be ecologically important in studies of extant taxa, but also other traits that just vary in anoles. And so I think it's a good representation of overall variation that also includes a lot of ecological traits. So the first thing we need to do to test this hypothesis is to estimate historical ecological opportunity, and we need to do that at nodes in the phylogeny. I took 2 different approaches for estimating opportunity. The first thing I did was I used an existing method whereby you simply represent ecological opportunity as assuming that it's scaled with time when a lineage diversifies. And so, this is fairly intuitive. If you have a diversifying lineage, the number of lineages accumulates through time, and therefore ecological opportunity should be inversely related to time. And so there are a few methods out there that rely on this basic relationship. However, there are a couple issues with this estimate of opportunity. I'll call this the time model. the main issue I was concerned with, especially relevant to anoles, is that it deals poorly with geographical heterogeneity in irradiation of a group of organisms. And so you can imagine that if you had this, the same radiation we had earlier, but you have colonization of a new region, then time is going to be a poor proxy for ecological opportunity. So we've got two different islands in this case, a red one and a blue one, and here are the nodes in the phylogeny. And time is going to overestimate the ecological opportunity at the red nodes. It's going to say, there's a high, because you're only using time as the proxy, you're going to estimate very low ecological opportunity, whereas in fact, you should reset the clock when you have colonization of a new region. You'll remember from the Anolis phylogeny that we did have diversification events in new geographical regions that were occurring later on in the diversification of anoles. And so we would underestimate ecological opportunity of these late-breaking radiations. So we developed a new model, which I call the Lineage diversity model. And it's a model that accounts for geographic heterogeneity and diversification. And so, so we'll walk quickly through it. For each node, we need two types of information. We first need to know what was the geographic region where that speciation event occurred. So every node is a speciation event. That's the first thing. And so if we have a node of interest, we first ask where did it occur. It's pretty simple. It occurred on the blue island. This is a very, very simple example. The next thing you want to know is who else was around. What else was on the blue island when that speciation event occurred? And it's very simple. You draw a line through the phylogeny and you count the number of branches that were on the blue island that cross this time slice. So if we go through there, count up the branches, we can say there were 4 other blue islanders. Now, this is a very simple dummy example, and no empirical system would ever provide you with such a clean example, of course. So in empirical systems, we have to deal with uncertainty in the geography of diversification. It's sort of a real bugbear for these types of studies. And so what we did with this method is we accounted for historical uncertainty using maximum likelihood. And so I won't go into too much detail on it, but what we do is we create for each node, so say we have this focal node Y, we create a lineage diversity estimate that's a weighted average of all the possibilities that node speciated on a given island. So here, according to this geographic reconstruction scenario, it's unlikely that this speciation event occurred on the yellow island, very low probability, and it's about an even split between yellow and blue. And so what we do is we count up all the yellow, we count up all the red and the blue, and we keep those tallies separate, and then we multiply them times the probability that it was actually on that island. And so we sort of create a weighted average for ecological opportunity. So it's still a node-based score, but it accounts for geographic uncertainty, and we measure that with a given model of geographic evolution. I will throw in a quick plug. The method is available on one of my co-authors in his R package, and he's blogged about it numerous times. It's a sort of a development blog, so you can check it out if you want. And the method does what we want it to do, essentially, for the Greater Antilles radiation. And in this plot, I have our new lineage diversity metric. These are all nodes in the phylogeny. Every data, every point is a node, and you can see that the colors correspond to nodes in the phylogeny on the left. And of course, we have pie charts where there's geographic uncertainty. And so we have time on the x-axis, lineage diversity on the y-axis, and you can see as time progresses, lineage diversity picks up, and then it resets when you have the colonization of Puerto Rico and Jamaica. And this is the variation this is the heterogeneity that we were trying to capture. And so it seems to make sense. So now that we've got our estimate of opportunity for those very same nodes, the same data points in the plot, we want to measure rates of evolution. This is a little bit more straightforward. There's already the math for this. So we can estimate rates using an independent contrast approach. And although Felsenstein wasn't explicit about this point, It's known, Liam Rebels discussed it in a couple of papers from the mid-2000s, that the variance of Felsenstein's contrasts are essentially a function of the evolutionary rate, assuming a Brownian model. And so you can estimate trends in rate, changes in rate by calculating independent contrasts and then measuring their variance as it changes with some other variable. And so what we did was, for each node in our phylogeny, we calculated the contrast for the trait of interest, and then we repeated for all nodes and for all traits, and then we fit the variance of these independent contrasts. We fit the relationship of the variance with the contrast to our ecological opportunity estimate, and that was the model comparison part. So now we have both of the kinds of data we need, and let's see what we find. So those are just sorry about that. Three models that we looked at: the time model, which is one ecological opportunity model, lineage diversity, and then in the third model, I'm assuming that the rate of evolution is constant across the Anole phylogeny for the trait. So what we find is we find support for the ecological opportunity model for two, the two main ecological traits in Anolis, which are body size and limb length. So I'll go through these one at a time. What we find here, so first, I'll talk about the graph. What I've plotted, remember everything's a node. Every data point is a node that corresponds to the phylogeny, and that the pie charts just represent geographic uncertainty. So lineage diversity is on the x-axis, and then body size contrasts, the absolute value of body size contrasts, are on the y-axis. And the dotted line is a fit of the variance of those contrasts. And what you can see is that the rate of evolution decreases as lineage diversity increases for body size. And this means that when opportunity is higher, when there are fewer competitors around, the rate of evolution is higher. And then it decreases with the saturation of island faunas.
Is that curve for the entire data set?
It is, yeah. It's for the, so
what happens if you take out Puerto Rico or Jamaica, which by definition don't have large lineage diversities?
So yeah, what we've done is we've just run a single fit basically because of the geographic uncertainty. Because if you it's hard to pull out individual islands because you'd be pulling out essentially fractions of some of these nodes. We're not certain where they went. We've thought about ways to do this, basically with stochastic character mapping. That's something we want to do in the future. To answer your question, I think if you look at Puerto Rico and Jamaica, the black and the green here, you can see I'm not sure that we have evidence for a decrease within those islands, but those islands have much lower diversities, both for island totals as well as for communities, than on the larger islands. And so perhaps they're not saturated with phenotypically. So we are considering the whole thing all together. Obviously, a lot of the pattern comes from the larger islands, but you do see there's a Jamaican contrast up there and a Puerto Rican contrast that are span a fairly big range. We get a similar. So this is body size, and body size is an important ecological trait. It's actually, before people talked too much about limb lengths with anoles, a lot of the early work in the 60s on anole ecology and niche partitioning was based on body size. It's clearly a way in which they partition resources. That's Anolis occultus from Puerto Rico. It's a subadult, but it's not getting much bigger than that. And then this is a Equestris from Cuba. It's one of the subspecies on one of the barrier islands. We find a similar result with limbs. In this case, we do fit a decreasing rate of evolution in limb length with lineage diversity, but it's actually the time model that's best favored. Both of these models are favored over the Brownian, Brownian model, but the time model is favored a little bit better than the lineage diversity one. And limb length is a classic ecomorphological trait. The anoles, anoles that use different microhabitats tend to differ in relative limb length. For the remaining axes, remember there were 3 other PC axes. The model results were ambiguous, and so head length, toe pad width, lamella number. These 3 axes explain a lot less variation in our data set, and I think there could be several reasons for this. They may not be evolving under the same ecological model, and it also, I tend to suspect that it also could have to do with sampling error. The fact that on these more minor PC axes, sampling error, you're going to have essentially a lower signal-to-noise ratio. That's just one explanation. We discuss it in greater detail in the paper if you're interested. Before I conclude this section, I want to talk about one other thing, which I'm sure several people have thought about, we're probably preparing questions on, which is what about extinction?
I was going to hold my tongue.
And obviously, extinction is a huge problem for the use of comparative methods for looking at macroevolution, and with good reason. It's simply hard to account for with phylogenetic methods. And I'm sorry to report that for all the fantastic things about anoles, one of the worst properties of the radiation is that there's just a really poor fossil record. I mean, everything that we know about the fossils of anoles, and it all basically comes down to a single lineage of green anoles from the Dominican Republic. And so we really can't say anything from the fossil record about anole extinction. What we did do was a sensitivity analysis of our methods to see what the influence of extinction is on these ecological patterns of trait evolution. And so very quickly, what we did was we took simulated phylogenies that were birth death phylogenies that had extinct taxa, which I've shown in red. We evolved them to 100 taxa, did this hundreds to thousands of times. And then we simulated trait evolution on these phylogenies under the 3 models that we were testing. So we simulated where the rate is a function of the lineages around at the same time, so that's a lineage diversity model. Then where the rate changes just with time, and then also just a Brownian motion model. And what we find is that power decreases a little bit with the lineage diversity model as you increase relative extinction to near unity, but it doesn't. You never get a shift. It's not going to misspecify as the most commonly favored model. So that was actually somewhat encouraging. Extinction affected it a little less than we were expecting. It's constant rate extinction, but it's still somewhat encouraging. With the time model, we have a similar pattern. The time model performs more poorly anyway, but extinction doesn't seem to be the most important factor. And under the Brownian motion model, we don't tend to overfit we don't tend to falsely fit ecological models when we add extinction. You can see that these, probability of type 1 error is relatively constant, even with high extinction. And so it's somewhat encouraging that if extinction is going on, it might not erase the pattern we're looking for. And so provisionally, I think the patterns we see are supportive of the ecological opportunity hypothesis. The 2 most important trait axes, we see these decreases in the rate of trait evolution as communities mature, or as island fauna saturate, I should say. So I'm going to shift gears quite a bit, actually, and talk about another project, which was sort of a fortuitous project. It's something that fell into my lap about 1.5 years ago, and I think it's kind of a fun story. I'm particularly excited to talk about it to museum folks. And I'm essentially going to ask how a classic hypothesis of convergence holds up in the face of a new discovery. It's a dinosaur type discovery. I'll start, however, with a little bit of background, just a little more on the anoles. So anoles are well known within biology and even outside of biology. And I think the thing that they're most famous for is the phenomenon of replicated adaptive radiation. And I didn't really talk very much about the replicated aspect of it with the previous study. But anoles are famous for producing similar sets of ecological specialists on different Greater Antillean islands. And so, specifically, anoles have produced these sets of ecomorphs on all 4 islands. And the sets of ecomorphs are quite similar. And so, ecomorphs are named for the substrate that they use. So you have the trunk ecomorph, which is found almost always on the trunks. And it has a morphology to do that. It's very spider-like, and it skitters extremely fast. You have twig anoles, which have short limbs and short tails, and they're diminutive little things that crawl along branches. They're very cryptic, they're very slow. They differ in their behavior and their ecology. And what's interesting is that these suites of specialists have popped up independently numerous times. So you get all 6 ecomorphs on the big islands of Cuba and Hispaniola. You get everything but the trunk anole in Puerto Rico. And then one less, you drop out the grass anole for Jamaica. So they're similar communities, or similar faunas. The term ecomorph is actually really coined to describe these anoles by Stan Rand and Ernest Williams. So Williams was really the person to make anoles a phenomenon. Ernest Williams, he was a professor at Harvard for decades. He had just an amazing group of graduate students. And it was this 1972 paper in the journal Evolutionary Biology that I think really started it all. And in this paper, Ernest really first addressed the similarity among evolutionary faunas head on, and he developed a model for diversification based on what he had seen in the Greater Antilles. So Williams had a deterministic model of diversification, and I think he was inspired to some extent by some of the deterministic models of community assembly that were going on in ecology at the time, and some in the very same department. What Williams realized is that he had examples of not just similar communities arising, but similar communities evolving in somewhat a seemingly deterministic fashion. And so Williams' model has been very influential. It's been modified much since its original conception, but it predicts that ecology really can affect the pattern of evolution. It can lead to the occurrence of it can lead to very similar outcomes. Now, a question you might ask is, where did the ecomorph concept come from? How did Williams come up with the idea? What are the sort of origins of his thinking on this? And I'll argue that there are 3 kinds of discovery that led Williams to come up with the ecomorph concept. And it really was, it was at least a decade in gestation. Here I've got it. So that's Ernest Williams to the right, and then his first grad student, first or second, I think there's some contention about that, Stan Rand. And they were really sort of the authors of the concept. So 3 kinds of discovery, I think, led Williams to recognize the anole ecomorphs. The first was that during Williams' time as a curator at Harvard, his lab oversaw the first real ecological studies of anoles in the Caribbean. And so it was actually Bruce Collette and Rudy Riebal, who weren't at Harvard, who did the first ecological studies. Bruce was mentored to some degree by Williams through correspondence. But it was really Tom Shaner and Stan Rand who made the ecological study of anoles an industry. And they published countless papers on the ecology of island anoles. And these guys and the folks that worked with them and the other students in the lab were really the first people to notice that the communities were structured in similar ways from one island to the next. And they actually studied mechanisms of niche partitioning. They studied competition. So this was going on during Williams' time, and really under Williams. So we had ecological studies and the discovery that we had similar things going on these islands. At the same time, we had a set of systematics discoveries that really put these ecological discoveries into a different context. And they were, I think, Etheridge deserves the earliest credit. His PhD thesis in 1959 showed that these ecologically and morphologically similar anoles from different islands actually weren't, each other's closest relatives. And it was only with these systematic discoveries that Williams and his colleagues started to gain the perspective that this really was a system where we had the convergent evolution of faunas. It wasn't assembly. Gorman and colleagues took it to the next level, essentially, with molecular studies and cytological studies. They really started to find out that groups that were long thought to be radiations actually were polyphyletic, and so this picture of convergence started to emerge. And there was one more type of discovery which rarely gets discussed, and it's the discovery of new species. And some of the seminal anole ecology papers from the early 1960s. It's actually funny to go back and read them because there's a paper on the community ecology of Puerto Rican anoles, and we hadn't even discovered all the ecomorphs on Puerto Rico yet. And so in 1963, in a 4-month period, these two species were discovered. And so this is Anolis occultus. It's a twig anole from Puerto Rico, and here's a twig anole from Hispaniola, Enceladus. Now, before this period of time in 1963, Puerto Rico and Hispaniola did not have twig anoles. They were on Cuba and they were on Jamaica. And so with the discovery of these remarkably convergent species, all of a sudden it sort of just smacks you in the face with this pattern. It's begging to be noticed, basically. It's really draws into clear relief the pattern of convergence among these islands. And so discovery, I think, played a real role. I will say that despite Williams proposing this deterministic model, he was very cautious about it, and he was always the first person to discuss limits to the model, drawbacks, areas where he didn't think it was a very powerful model. So I think the thing that concerned Williams the most was that not all Greater Antillean anoles could be classified as ecomorphs. And, he sort of had two categories of Greater Antillean anoles: those that really fit the bill and those that were inexplicable. And so, the distinction here is that, there are a lot of anoles that can't be, even in the Greater Antilles, it can't be classified as ecomorphs. And what makes it an ecomorph is if it's arisen numerous times independently. So that's what suggests a deterministic element. However, if you go to the Greater Antilles, you get things like Anolis bartschii, which is a rock wall specialist on Cuba, and it has no analog. If you go to Jamaica, Puerto Rico, Hispaniola, you don't get a rock wall specialist. This thing clearly seems to be well adapted for what it does, but it hasn't arisen numerous times. So it's sort of a strike against the model. Another example would be the Cameliolis anoles from Cuba. These are these giant, slow, cryptic anoles that live up in the canopy, but are the size of crown giant anoles. They're huge. They're totally bizarre. These, they're so distinct that they were actually given a different genus for for over 100 years, called Cameliolis. They were thought to be basal to anoles. And it's nothing like them on any other islands. So the model didn't explain everything. After the 60s, after Williams' 1972 paper, there were still anoles to be discovered, but that would change about a decade later. So there was sort of this period where species were continuing to be discovered, but at a decreasing rate. But with each new discovery, it offered sort of a test of Williams' model. And so when Anolis fowleri was discovered by Al Schwartz in the 70s, this is another unique anole, so that's kind of a strike against Williams' model. It's like, oh well, this doesn't really fit the prediction that things should match across islands. There's nothing else like it. However, when Chaplainai and Guazuma were found in later years, they matched twig ecomorphs, but represent independent evolutions of it. So these, this is new data actually allowing a test of a model. After 1984, though, there's been no discovery of a Greater Antillean anolis of ecomorphological significance, let's say. There have been new discoveries of species, but they've always been cryptic species. You split something up, you know immediately what it's related to. So for 25 years, we haven't had the discovery of novel forms, basically. And that was essentially the situation until March of 2010. And so March of 2010, I was teaching a herpetology class with Jonathan, and I checked my email one morning, and this fellow, a colleague of mine named Miguel Landestoy, he's a fantastic naturalist, had sent me an email. He said, I caught an anole last night when I was out looking for birds, and it looks like Camelliola's from Cuba. It's unlike anything I've ever seen. And every once in a while, I'm sure there are a lot of systematists who can sort of relate to the occasional proposition by someone, hey, I think I've got a new species. And you're like, okay, yeah, sure. I'm sure I'm not the only person who's hiked for 2 days to find something that wasn't a new species. So I sort of dismissed it, and I said, send pictures and then we'll talk. And I went off and I did lab, and then came back from lab, and then these were in my inbox. I had these pictures of this anole, and I think most people in the room probably know what I'm talking about when if I were to say there are two kinds of new species. There are new species where you're like, hey, it might be a new species. Check under its legs, see if the scales are different. And then there are new species where it's like, what is that? That is something. And this immediately, new species that's unlike anything on Hispaniola. So I was ecstatic. I called Jonathan in to take a look. I quizzed him. I said, what is it? I got this picture. I didn't tell him it was a new species. He's like, where's it from? It's from the Caribbean, new species. He's like, immediately. It's obvious. So I did two things when I got this email. First thing I did was I checked my schedule, and it was bad, but it was getting worse. It was like a 2-day window, 4 days later, and I checked how much money I had left on my dig. 5 days later, I flew down to Santo Domingo to go get this thing. And basically got the first flight that was within my price range. And motored down for a 36-hour trip to the Dominican Republic to grab this new anole. I've worked for a number of years in the Dominican Republic. We have amazing colleagues and collaborators down there. Jorge Broca from the Ornithological Society managed to get me an ultra-rushed export permit. So I got down there. Miguel, who had caught the anole, met me in Santo Domingo, showed me the anole, and I took a look at it. It was a female, unfortunately. And if you're going to describe an anole, you really want a male. And I had 20 hours left, and he assured me that the site was only about 6 hours away. So, hey, let's go see if we can find some more. And so we jumped in a truck and took off. We crossed our fingers that A, we'd get there before dark, and B, the weather would hold out, because it's up, sort of in mid-elevation mountains. For those who haven't been to the Dominican Republic, it's a gorgeous island, some of the scenic drive. However, as we approach the foothills of Marucos, things started to change a little bit. It's getting a little stormy. The terrain gets a little rougher, going gets a little slower. Of course, as we start getting closer, it starts to rain. This is what it's looking like as we're sort of approaching the closest town. And then I took this picture to show that it was raining pretty hard, but I'm not sure you can tell. But we were undeterred. We had very bright lights that I had charged up the night before, and we looked around till 3 in the morning, and we got 2 specimens, including a gorgeous adult male. And so I present, for the first time outside of my graduate institution. Story's not over yet, forgot. Got back in the car, drove all the way back across the island, got some pastries from Miguel's mom, and jumped on a plane. This is Anolis Lizards, which description is in my thesis and about to submit it for publication. It's a new and bizarre giant Anolis from Hispaniola. And it's only found, I've been down a couple times since, it's only found in this one location, which is basically in the northern foothills of the Baruco Mountains, right on the border with Haiti. And it's sort of a dry scrub forest. It's found with Spanish moss is very lichen-covered habitat. Here's what the scalation looks like. Scalation actually is indicative of its relationships, although we had no idea when we first looked at it what it could possibly be related to. And so, yeah, what we wanted to ask was, we have this new species. This is the first time we've had an opportunity like this since the mid-1980s. So Williams has a deterministic model, and we wanted to ask basically, is Anolis landistoi ecomorphologically similar to another ecomorph or other Greater Antilles anoles, and what's its closest relative? And so I'll go through this part fairly quickly. So we did an ecomorph. We quantified morphology basically by measuring the 11 classic ecomorph traits, and it's a subset of what I measured earlier. But all the ecomorphological studies on anoles have looked at this trait set basically. So we calculated Euclidean distances in morphic space to find its nearest neighbor. What's it most similar to? And then we simply generated a phylogeny to find its closest relatives. We used both mitochondrial and nuclear DNA and did a lot of different analyses to look at this. So as for what it's most similar to, Miguel was right. It's most similar to these bizarre Cuban anoles, hitherto non-ecomorph anoles from Cuba. So what we're calling giant twig anoles. And these are Euclidean distances, and this is a frequency distribution. So you can see that it's closest to all of the species. And also some giant anoles and twig anoles, and then the remainder. And so it is quite similar to this lineage from Cuba. So here's just another morphospace plot, and I've just plotted a couple of the key axes. These are the same axes from the previous study, where you have essentially limb lengths and then body size. And you can see that the new guy sits up here, sort of closest to that cluster with Cameliolis. I'll show you other axes as well in a second. So it's a close match. So now, of course, we want to know, is this a close match because it's a sister taxon, or is this an example of ecomorphological convergence?